So, bon dia, bon dia. So, with a little bit of delay, I guess the test tournament was a success yesterday, and everybody stayed a little bit later, and then who won? Somebody knows? You, yeah, and did you win? Okay, so I gather with that. So we start with the flash talk session, and I get it that for the moment only Jaime is here. Jaime is a PhD student in my group, and so I give the word to Jaime Sien, who is also the coordinator of the informatics department at the moment, and so.
It's fine. Hello, everyone. I'm talking about nectar. It's like tools is my PhD work, and so nectar is a platform or framework or application. The intent as the completely toolbox for network analysis. I can build, like create a network with the electrophysiologist's raw data. So the platform has there is some strategy or tricks for the could be analyze this data and create these networks and do some analysis in the in this fluxogram, you can see how can this tools works. And I'm load data and spikes. Normally we can use spikes and FLP data for do matrix correlations and expect coherence calculator. And the partitional direct coherence for the flow for this data. And there is the tool has an engine for pre-calculate all these things, these stuffs, and create a new database from this data. And there is a dashboard for present this analysis with the graph forms, and the particular feature in these tools is you can analyze only in the static form for this network, but I think it's possible to analyze the dynamic connectome and in the time is like a D-norm. So this platform can build mix network analysis with using small worldness or another metrics. And make a graphic for all the on the slide windows like this, this kind of the schematic view, or on the make networks and relating the region, the interesting regions in the brain. And do not a lot of analysis for the neuroscientists, for the make interesting insights for the studying this. If you interested about my results, I invite you for the look my poster on the upstairs. Okay. Okay. Oh. Okay. So I think this we can talking about these results. I think the short presentation has no space for elaborate for this, but I think this is a good question. But I have some clues for the. It's not a. Just there is a let me see. I have some clues for this data makes sense with this when looking the others, the other resource and the others student is there is okay. Yeah. Okay. 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 Yeah. 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 Okay.
Hi, everyone. Good morning. We are very happy to have you here in our second day of the House Symposium. We are beginning now the session, Disrupted Pathways. And I am Caterine. I am a PhD student here in the Institute. And our first guest is Professor Marcos Costa. He is an expert in cellular reprogramming, cell therapies, and the use of advanced in vitro models, such as human-induced pluripotent stem cells, for studying the pathogenesis of disease like Alzheimer's and other forms of dementia. Professor Marcos spent a few years outside of Brazil doing his researches and working in the pharmaceutical industry as a lab head. But now he's back. He's back as a professor here in the house. And please, Professor, the word is yours.
So good morning, everyone. Thanks, Catherine, for the kind introduction. So as Catherine just said, I'm back to Brazil after some years. And the work that I'm going to present today was performed in my lab at the PRED Center in Roche, where I was working the last 2 years. And it's mainly focused on modeling TDP-43 loss of function in human-induced neurons. And I will try to explain why this is important. So ALS, amyotrophic lateral sclerosis, it's a fatal disease that happens in between 6 to 9 people in 100,000. So it's considered a rare disease, but it's a devastating disease that it's mainly sporadic, so we don't really understand what is the cause. We know there are some genetic risk factors, but we don't fully understand why the disease happens, but as I will show later, there are some hints. And there are about 5 to 10% that are familial cases that are associated with specific mutations, such as in proteins such as C9orf, SOD1, TDP-43 or TRRDBP, that is the name of the gene, and FUS. It leads to a progressive weakness in muscles, and the region that it's the body that it's more affected, the upper limbs, the lower limbs, or the bulbar region, depends a lot on the region of the spinal cord that the disease happens first. So the clinical manifestations, they are highly heterogeneous. But regardless of how it starts, most patients will die between 3 to 5 years after diagnosis, and most of them between 2 to 3 years. So it's a very fast progressing disease and with a very deadly outcome. So what do we know about the pathology? What can we see in both familial case, or most familial case, and sporadic, that could help us to get some idea what is the cause of this disease? So one common hallmark of the disease is the so-called TDP-43 pathology. So TDP-43, as I mentioned, is the protein encoded by the TRDBP gene that is the transactive response DNA binding protein. It has 43 kilodaltons, that's why it has this name. As the name also suggests, it binds to DNA, but it also binds to RNA. So it's actually more considered as a classic RNA binding protein. And as such, it has several functions in regulating splicing, our transcription, RNA stability, RNA export, and also microRNA stability. Yeah. It's good. And when we study the brains or spinal cords of patients post-mortem, we observe that in 97% around of the patients, there are so-called cytoplasmic inclusions. So this is what is shown here. We see in the motor neurons, this is the spinal cord, but we also see in the primary motor cortex. We see this accumulation of TDP-43 in the cytoplasm and the formation of aggregation. So pretty much resembling what we see in other diseases, but for other proteins, right? So if you look Parkinson, we have aggregates of alpha-synuclein. In Alzheimer's disease, we have aggregates of hyperphosphorylated tau. And ALS, the hallmark would be the aggregation of TDP-43 protein. That it's also modified, there is also phosphorylation, ubiquitination. There are several modifications in the protein in the aggregates. Another consequence of these aggregates in the cytoplasm is that it leads to a depletion of the nuclear TDP-43. And therefore, because TDP-43 is depleted from the nucleus, all those functions in regulating transcription, splicing, that happens in the nucleus are supposed to be affected. And this, of course, could lead to several changes in the gene expression because TDP43 would be losing its normal functions in the nucleus. And indeed, in the last years, there has been a huge amount of effort to understand, especially this step, the so-called alternative splicing regulation, that it's mediated by TDP43, and how the change in splicing elicited by this TDP43 loss of function could be affecting disease. So the hypothesis is that when you have the aggregation in the cytoplasm and the depletion of TDP43 in the nucleus, this would lead to changes in gene expression or splicing alterations, and that would be the cause of neurodegeneration. And of course, this is a mechanistic proposal, and based on that, there have been many drug discovery programs, and there are some actually already in clinical trials, for example, trying to correct the splicing of some TDP43 targets. There's one by Corrales that we will have the data sometime next year, that it's focused on a single target, that it's statin 2, and the claim is that by correcting the splicing, this single target would be sufficient to rescue neurodegeneration and therefore bring some clinical benefit. We will need to see. Yeah, so this is again bringing this hypothesis. So when the loss of nuclear TDP43 function would drive neurodegeneration, the alternative hypothesis is that it's the aggregation in the cytoplasm that would lead to a cytotoxic effect, but I'm not going to talk about this today. And as I mentioned, this would be related to splicing alterations of hundreds of neuronal transcripts. So this is a scheme just, again, representing the rationale of this hypothesis. So in the health condition, TDP43 is regulating RNA splicing, RNA transport stability, and so on. And in the ALS patients, but this is also observed in some frontotemporal dementia case and also in some regions of the brain, Alzheimer's disease. The loss of TDP43 in the nucleus would lead to change in gene expression and mainly the change in alternative splicing. Here I want to highlight one important event, that it's the inclusion of what we call a cryptic exon. So usually these cryptic exons, they are skipped during the splicing event. And when they are included in the absence of TDP43, when these cryptic exons are included, this very commonly leads to a nonsense mediated decay of the RNA, because you have a change in the read frame of these transcripts, and therefore this RNA is not translated anymore, right? And as a consequence, the inclusion of this cryptic exon leads to the depletion of the protein that would be encoded by this RNA. Yeah, and this is basically what is depicted here. This is a paper that was published last year, where they analyzed these cryptic exons, I mean the inclusion of these cryptic exons in different genes across different brain and spinal cord regions in patients, and also in different cell models. And we see here that there are many events that are specific for ALS and FTD. So what we wanted to do here was very simple. When we are developing some drug discovery model, ideally we want to have a simple model that captures some aspect of the disease, and in this model we can test thousands of compounds, thousands of drugs that could potentially impact this specific mechanism that we are aiming to. So the idea was, if the TDP43 loss of function is really important to affect gene expression, splicing, and neuronal properties, we should be able to capture that in a simple model. So we wanted to basically design an experiment where we could knock down TDP43 to a certain level and investigate the impact in gene expression, and also some readouts that are resembling ALS phenotypes. So neurodegeneration, neuronal hyperexcitability, and so on. So our design was very simple. We cultured glutamatergic neurons or cholinergic neurons, that here we are calling motor neurons, because that's the name that most people give to these cells, although I don't like. I would prefer calling them cholinergic neurons, but we have them separated. So either we have the glutamatergic neuron cultures or the cholinergic neuron culture. Why this is important? As I mentioned, this is a motor neuron disease. In the spinal cord, motor neurons are cholinergic. In the motor primary cortex, they are glutamatergic. So the upper motor neurons, they are glutamatergic, and the upper motor neurons, they are cholinergic. We wanted to understand if they would be equally affected by TDP-43 loss of function. To knock down TDP-43, we used antisense oligonucleotide. So it's a small sequence of DNA, RNA that binds to the RNA and leads to its degradation. And therefore, we can knock down our target using this sequence. We treated the cells after 4 days, and then we followed these cells in culture up to 5 weeks using multi-electrode array electrophysiology to record neuronal activity. And at the end of the experiment, we fixed the cells to do different immunocytochemistry, RNA sequencing, and also viability assays that I will show at the end of my talk. So very quick parenthesis here, just to convince you that these glutamatergic neurons and cholinergic neurons are indeed what they are supposed to be. So these are just an image showing the morphology of the cells in culture, neurons labeled with the microtubule-associated protein, MAP2, that it's a pan-neuronal marker, or this neuronal marker AGUCD. So you can see here the overall morphology. And in these glutamatergic neurons, we also see that the cells express the glutamate transporter VGLUT2. In the motor neurons or cholinergic neuronal culture, again, the same markers, but here we see some key markers of cholinergic neurons, such as ILET2 and the acetylcholine transporter chat. We also performed RNA sequencing of the cells, and as expected, they are very different. When we compare glutamatergic neurons to motor neurons, we see that in the motor neurons, we have a huge increase in the expression of genes associated with cholinergic neuronal phenotype. And indeed, when we do gene enrichment analysis here, we see that the genes that are upregulated, they enrich for neuromuscular junction. So confirming that we managed to have some neurons with molecular features of cholinergic cells in vitro. And here, again, some classical markers of glutamatergic neurons, the glutamate transporters 1 and 2, and here, again, transporters of acetylcholine. As I mentioned, we also use multi-electrode array recordings. So very briefly, methodological here. We use the Axon Biosystem machine with a multi-electrode array plate, where we basically have multiple electrodes in each well, and we can culture cells in all these different wells in different conditions. We record the cells in this equipment, and then after, we do a spike sorting to identify the activity of specific or unique neuronal cells, and this is basically the data that we will later analyze to identify firing rates, bursting firing, neuronal network activity, and so on. And the first thing that we can clearly see here is that when we compare glutamatergic neurons and cholinergic neurons in culture when they are alone, here they are co-cultured with astrocytes, but the only glutamatergic neurons or glutamatergic or cholinergic neurons, we see a clear difference in the network activity that these cells are able to establish. Probably not very surprisingly, considering the nature of the glutamatergic transmission, the cholinergic transmission in the nervous system. But we see that these cultures here, they manage to establish very nice oscillatory activity after 3-4 weeks in culture, whereas the cholinergic neurons, they are firing, they are active, but they fail to establish the synchronized activity. So basically, here we see a raster plot of neurons in the same well, and here is the array density spike representation of the activity in the same wells. So we see here that in a population level, we have a very clear synchronization of the cells in the culture, but this is not observed in the cholinergic neuronal culture. And one feature that we saw that can help to mathematically identify this difference is what we call the ASDR skew, that it's mainly how different from the the baseline, we see these peaks in the SDR activity. But we also see that when you look classical cross-correlation, for example. So again, there is a strong cross-correlation in the glutamatergic neuron culture, but not in the motor neurons. And this will be important to understand the results that I will show later. Okay, now back to our main question. So we first wanted to characterize what is the degree of TDP-43 protein knockdown that we have in our models. So again, just to refresh your memory, that is the experimental design. We treat the cells with the TDP-43 ASO day 4. And here we are looking at the TDP-43 protein expression at 5 weeks, 35 days. But we also look this at 21 days, so the protein is gone after 2 weeks, to make a long story short. But this is 5 weeks. So you see here that in the control condition, TDP-43 is strongly expressed in the nucleus. After knockdown, we see a clear reduction in the signal in the nucleus. And when we quantify that, it's more or less a 50% reduction. We also confirmed that using ELISA to measure in a more quantitative manner, and it's more or less between 40–60% knockdown. So we have about 50% reduction in TDP-43 expression in both glutamatergic and cholinergic or motor neurons culture. After RNA sequencing, we see that RNA depletion, it's even a bit stronger. We see about 20% only compared to the levels in the PBS, and the non-targeting ASO is not significantly affecting TDP-43 expression. So we have a fairly successful way to knock down TDP-43. I'm not showing here, but depending on the concentration of ASO we use, you can also play with the knockdown level. So if this is 5 micromolar. If you use 1 micromolar or 0.2, we see a weaker knockdown of TDP-43, and this is also interesting because we can now use this data to see how is the dose response of the genes that are affected by TDP-43 loss of function. So what is the impact of TDP-43 knockdown to gene expression in these cells? So these are volcanoplots for motor neurons, and glutamatergic neurons, showing genes that are significantly downregulated or upregulated upon TDP-43 knockdown. And we don't need to spend a lot of time in each of the gene here, but what is important is that we can recapitulate the downregulation of key TDP-43 targets. I mean, like statamine 2, for example, that I mentioned earlier, that it's being pursued clinically, but also it's very important for axonal stability. So that's why it's a target that many people believe to be important, because it's an important protein for the maintenance of the microtubule in the axonal process of neurons. Kinase 2B, that it's also, don't need to spend much time here in this audience, but it's a key kinase downstream of calcium signaling in neurons, regulating many important functions for gene expression, neuronal activity, and so on. Anki13B, that it's an important synaptic protein. ATG4B, that it's associated with autophagy. So all these proteins very well described in ALS patients and considered to be key targets of TDP-43. So all these genes we can recapitulate in our models. We also see that there's some overlap, but we also see some genes that are only affected in motor neurons or only in glutamatergic neurons. So it's not that both neuronal models show exactly the same gene expression change, but many are overlapping. So, okay, gene expression is interesting, but as I mentioned, TDP-43 can regulate gene expression directly by regulating transcription, but also by regulating splicing. And as I also mentioned, the splicing mechanism is considered to be one of the key mechanisms in the pathogenesis. So we wanted to more directly look for changes in alternative splicing. And the way we do that, it's very simple. So if you look here, this first scheme, when you have the pre-mRNA and you have, for example, the inclusion of this exon here, you have a variant of the gene that we call variant 2. If you have this exon being skipped, that it's usually, as I mentioned, what happens for most TDP-43 targets, you have the variant 1. So in the normal condition, we would mainly have the variant 1. In the pathology, because TDP-43 is depleted, we would start to see the variant 2. So by calculating the frequency of these two variants, we can calculate this VF index, that it's a way to measure how this alternative splicing product is being represented in different models. And then when we do that, again, for motor neurons or glutamatergic neurons, we clearly see here several splicing events that are changing the frequency in both models upon TDP-43 loss of function. Again, we see here that the stathmin-2 product with the cryptic exons, it's strongly upregulated. So this fits very well to the downregulation of the gene, right? So you have more stathmin-2 RNA with cryptic exon. This RNA is degraded, so less stathmin-2 in total. Again, we also see CAM kinase 2B. We see some other targets. And here, just to summarize what we see in numbers, we again, we again find many events that are shared in the different cells, but we also find a lot of events that are unique to each cell type. So this suggests that depending on the gene that we want to study, we need to choose very well what's the neuronal model that we are going to select for those studies. And here's again a comparison of how these change in gene expression and splicing can overlap, and therefore could the alternative splicing could explain the change in gene expression. Very briefly here, I mentioned that our multi-electrode array recordings, electrophysiology recordings, they are performed with astrocytes in the co-culture. So we just wanted to make sure that adding the astrocytes would not significantly impact the alternative splicing that we see. So we did the same sequencing in the presence of astrocytes. And so here we see the neuronal specific events. They are pretty much preserved in the presence of astrocytes. So having astrocytes do not affect anything on the TDP-43 loss of function impact in neurons. But interestingly, we also see some events that are astrocyte specific. And one of them, this ADGFL2, was very interesting to observe, because this has been suggested 2 years ago as a potential biomarker for ALS. So people identified this cryptic, in this case we have the cryptic exon and the formation of the cryptic peptides. So people detected these cryptic peptides in the CSF of ALS patients, and there are some efforts to characterize this as a biomarker. But what this here shows is that these, although interesting as a biomarker, if ever confirmed, it's not a neuronal TDP-43 target. So it would be a biomarker that is not related to the neuronal pathology directly. Yeah, so as I mentioned earlier, I mean, we know we have this information about splicing chains in ALS samples in the spinal cord and in the frontal cortex, for example. So we wanted to compare if the molecular signature that people find in patients' sample are recapitulated in our motor neurons and glutamatergic neurons. So we also took some publicly available data in another neuronal model in vitro. And yeah, just make a long story short again, what we see here is that the patient alterations, they are recapitulated in our models in vitro. However, there are many events in our in vitro systems that do not seem to be observed in the patients, especially in the spinal cord. So here we don't know if it's a power issue or if simply of the analysis, or if the motor neurons in the spinal cord that are a very minor population, they are gone when people do the RNA-seq in these post-mortem samples, or if this is simply an artifact in vitro. But what is good to see is that at least the events that are observed in patients, they are recapitulated. So we could propose that at least for the impact of these genes that seem to be relevant in the pathology, we could study their functions in these in vitro models. Yeah, and then here's just color coding showing the things that are similar to the frontal cortex or the spinal cord. Okay, now in the last 5 minutes, let's look a little bit what is the functional consequence. Okay, so I basically try to convince you that by knocking down TDP-43 in about 50%, we can elicit change in gene expression, alternative splicing that resemble change that we see in ALS patient sample. So what would be the functional consequence? Is this affecting neuronal properties? Is this affecting neuronal functions or survival? Because of course, if you want to test something as a drug that would increase neuronal survival, you first need to make sure your model can elicit change in neuronal survival. So we analyzed our electrophysiology data in different time points. I'm just showing here the 5 weeks time point. And what we see here, these are again, we discussed it before, right? The difference between glutamatergic neurons cultures and motor neurons. But what we can already see here in this representative raster plot is that when we treat the cells with the TDP-43 ASO, we see a clear increase in the frequency of these population peaks, right? I mean, the increase in the number of times that the neurons show this collective activity in a well. And this was very surprising for us because, I mean, most people think that, for example, when you remove TDP-43, you have an impact in UNC13A, UNC13B. These are synaptic proteins. So the expectation would be that you have a reduction in the synchronization, not an increase in the synchronization. But yeah, that's what we observed. And another feature that many people also describe, and this is a feature of many neurodegenerative disease, not only ALS, is the so-called neuronal hyperexcitability. And we also observe that we see an increase in the firing and the burst firing frequency upon TDP-43 knockdown. In the motor neurons, network activity doesn't make much sense because, again, they don't form a clear network. But what we also see that it's similar to what we observed in the glutamatergic neurons is the increased firing frequency and burst frequency. So intrinsic neuronal properties seem to be equally affected in both neuronal types, but the network impact, it's probably not possible to study in the motor neurons because they fail to form a proper network. And indeed, when we look these populational networks, and I'm not going to details, but we have about 26 different metrics that we can extract from the population effects in using cross-correlation or the SDR-derived metrics. So we put all these different features in a PCA, and we try to see how they can help to distinguish the conditions. And we can nicely see here that in the glutamatergic neurons, our PBS or non-targeting ASO-treated, they are close together, and the TDP-43 knockdown group is clearly different. However, in the motor neurons, we cannot separate the impact of the treatment using this more unbiased approach. Finally, we wanted to see if the neuronal survival would be affected. And then for that, we tested cell viability using different assays, such as BCA assays, cell glow titer, caspase. So there are different assays. I'm just showing one of them because they all did not show any change in the baseline. So TDP-43 knockdown is not sufficient to decrease cell viability under basal conditions. We also checked NFL release. So this neurofilament light chain. This is one of the main biomarkers for ALS. It's a distal biomarker, but it's clearly increased in the CSF and plasma in ALS patients, and probably because the axons are degenerating and releasing this protein in the fluids. Again, no difference, even some weird, but probably one sample here bringing this increase in the motor neurons, but no decrease, no clear increase in the NFL. And by immunocytochemistry, again, neurite length, so neuronal morphology, again, thinking about the role of statin 2 in axon process and so on. No big change, maybe some some trend in the motor neurons, but not significant. And when we look the proportion of neurons astrocytes, only here we found some drop in the ratio neuron astrocytes in the motor neurons, but that was the only change. So at the baseline, knocking down TDP-43, 50% doesn't seem to make neurons less healthy. So can we push this a little bit, putting some stressor? So as I mentioned, there's neuronal hyperexcitability. It's a very important process in neurodegeneration. So we can mimic that by adding glutamate. So we have a glutamate excitotoxicity in our cultures, and we can see how this impacts cell survival. And I will not go into all the data here, but just to show you, I mean, in increasing concentrations of glutamate, we can clearly see a drop in the cell viability. So we can induce neuronal degeneration by treating cells with glutamate. And this can also be clearly see here when we compare PBS and glutamate 10 millimolar. So we see that we've mapped to a reduction in the neurons and or the size of process of both in the cultures. However, neither in the motor neurons nor in the glutamatergic neurons, the knockdown of TDP-43 changed the impact of glutamate. So the neurons died in both conditions, but they didn't die more in the TDP-43 knockdown. So again, this would suggest that knocking down TDP-43 is also not making the cells more vulnerable to the hyperexcitability. So the conclusions is that regarding gene expression splicing, we can recapitulate most of the change that we see in ALS samples in an in vitro system. We can observe also some impact in the electrical functional properties of the cells, and thinking about neuronal network, mainly in the glutamatergic neuronal cultures. But surprisingly, we don't see any change in the neuronal vulnerability in neurodegeneration. And this was really something that surprised us, because if you cannot see that, we cannot rescue, right? So there is no meaning to, we see the change in gene expression, but there is nothing. So this could also raise the question if it's really the loss of function of TDP-43 that it's leading to neurodegeneration, or if you should consider other mechanisms or the gain of function that I mentioned earlier, that the aggregation in the cytoplasm, as the main driver of neurodegeneration. Yeah, so yeah, just to thank the people who did the work. So this was mainly performed by Vera Gizin-Filippa, who was an intern in the lab, and the 3 associates who helped with the cell culture experiments. Karsten, who contribute a lot for the analysis. Dwight Newton, Brad Friedman from the Gentech, that's now GRED, and did the patient data that I showed before. And thank you for your attention. I'd be happy to take questions.
Thank you so much for your amazing talk, Professor. And now we have time for some questions or comments. Does it?
Good morning. Nice talk. I just have a question about your cholinergic neurons in the cultures. So are they motor neuron? Are they also releasing glutamate like normal motor neurons? You know, motor neurons will release acetylcholine on the motor end plate, right? But also locally inside the spinal cord release glutamate. So do you have that dual phenotype, or do you have only acetylcholine release?
We don't know. We did some pharmacology for blocking AMPA and then the receptors in these cultures, and usually what we see in the glutamatergic neuronal culture is an impact in the synchronization. But because these cholinergic cells, they fail to show that, we didn't see any clear difference, but we saw some change in the firing rate frequency. So there could be some glutamate release, but that is not sufficient to establish a clear network activity. But yeah, we would probably have some, and we see genes of related to glutamate transport also there. It's just very low compared to the glutamatergic neurons.
Yeah. Would it be possible to grow some kind of interneuron to mimic the rancho cells? Is that possible?
Yeah,
Because
it's
then
possible.
you might get a
Yeah.
completely different phenotype.
Yeah, it's possible. We have a different model that where we combine glutamatergic neurons, these cholinergic cells, in a kind of sphere, and we also combine that with a muscle sphere, and it's very nice because we can use we see the muscle contracting, and we can actually use optogenetics to stimulate the neurons and see contraction. It's very beautiful, though very complicated for drug discovery because it's highly variable. So it's difficult to have two spheroid systems behaving exactly the same.
Hi, Marcus. Nice talk. So your data is showing that probably the direction of protein aggregation may be more related with the viability of the cells, right? From the clinical studies in genetics, from the mutations in the TDP, what is the fraction of the mutations that are more related with reduction of the function and with the aggregation? And another question is, if TDP is a general splicing regulator, that would be very strong the effect of reducing, right? Because it would affect many genes, as you show.
Yeah, these are good questions. The first one, for the familial cases where there is some TDP43 mutation, they are mainly in the C-terminal domain. And the C-terminal is the intrinsically disordered domain, so probably more associated with the aggregation behavior of the protein. We did test the impact of some of these mutations in iPS cells, and we did not see any impact in the splicing change. So again, speaking against the idea that these mutations are affecting the function of TDP43 as a splicing regulator, it should be more inducing aggregation indeed. The second question is about the impact of TDP43 in splicing. So it's not a general, I mean, it's usually binding to GT stretches. So these splicing junctions, usually they have GT repetitions. So TDP43 has about 1,000 potential targets that are described by ClipSec data. So these would be the potential targets. It's still a lot, right? I mean, 1,000 potential targets is a lot. But usually what we see in our models in patients, it's more in hundreds. So probably many of these targets, they are probably in non-coding regions. They may have effects, but other effects that it's not directly associated with splicing. Yeah, so it's probably disease relevant. We are probably talking about some hundreds of targets. But anyways, it speaks, I'm not very, I hope I'm happy to be wrong, but I don't think that rescuing one single target will make the difference. But I mean, I hope the best for the patients and the company, but I find unlikely.
Thanks, Marcos. So in theory, you are, along with the production of these splicing variants, you're also producing, so the neurons are also producing defective proteins, right? But apparently they are dealing well with this production of proteins that have no function, right? Have you checked if they are overexpressing like proteasome, like the digestion of these proteins, so they don't accumulate these proteins in the cytoplasm?
Yeah, well, so for most of the splicing, when you have these cryptic exons, most of them, they lead to out-of-frame readings, so you have nonsense mediated decay. So this is not translated, so you don't have protein products. Or just a few that are still in frame, and then you have a cryptic peptide. But the accumulation of TDP43 in the cytoplasm is associated with changes in the proteasomal system, in the also in the UPS, I mean the ubiquitin proteasomal system, and also lysosomal degradation. So there's another hypothesis for explaining the disease is that actually you have a failure in this protein clearance system, and that's actually leading to the accumulation of TDP-43, especially misfolded TDP-43, and then the aggregation. So there are some drugs that are targeting only this aggregation side of the coin, and there are some other approaches focused on the loss of function. Unfortunately, it's very difficult to study both together, and that's usually why people are approaching both mechanisms separately.
Professor, thank you so much once again. And now our next guest is Bruna Lorena. Bruna, she is a biologist, and she's doing PhD here with Professor Eduardo Siqueira.
Hi, good morning. I'm going to give a presentation in português. Nosso trabalho ele é intitulado A influência da infecção vírus Zika na maturação do sistema GABAergico cerebral. Meu nome é Bruna, né, como a Catarina falou, sou aluna de doutorado do professor Eduardo Siqueira e co-orientada pela Catarina Leão. Falando um pouco sobre o contexto histórico, o Zika vírus está completando agora a síndrome congênita 10 anos do seu acontecimento. Os dados mais recentes que a gente vê em relação Brasil, que a gente vê de 2015 até 2023, sobre a quantidade de nascidos confirmados com a síndrome congênita do Zika vírus, que é aquela que causa a microcefalia nos bebês ainda no útero da mãe, após serem picadas pelo mosquito da Aedes aegypti, que causa a Zika. Teve uma taxa de nascimento em 2015, 2016, que foi o auge, com mais de 1.600 crianças. E ainda continua acontecendo, embora não seja tão notificado. Porém, os casos, para eles serem realmente confirmados, demoram um bom tempo, e a gente sabe que os boletins epidemiológicos demoram um pouco a sair com essa confirmação. Mas até 2023 já tinha sido confirmado mais 71 nascimentos com a microcefalia congênita. Em relação ao número de óbitos, a gente teve uma quantidade que ainda acontece nas crianças após o nascimento, devido às sequelas, mas nos primeiros anos, 2015 e 2016, foi 48 e 121, e por municípios de residência já foram 47 até 2023, de 2018 a 2023. Uma característica das pacientes que possuem microcefalia, por causa do Zika, é a presença de epilepsia em boa parte desses pacientes. Esse artigo ele mostra bem isso no estudo feito em São Luís, com os pacientes de lá, onde eles avaliaram 109 pacientes, e desses, 68% tinham crises epiléticas, enquanto cerca de 9 não apresentavam, ou 32 não apresentavam. Em relação ao tempo, né, de quando eles apresentaram a primeira, ainda são com meses de vida, mas uma característica muito importante sobre essas epilepsias é que ela é durante o desenvolvimento embrionário, o nosso principal neurotransmissor inibitório, o GABA, ele é excitatório durante o desenvolvimento. E após o nascimento, quando tem a maior expressão, né, que vai se desenvolvendo o KCC2, que é o que vai colocar o cloro para fora da célula, quando tem esse maior desenvolvimento, passa a ser um sistema inibitório, o GABA. Porém, algumas situações durante o desenvolvimento podem fazer com que essa reversão seja inibida. E aí você vai ter um sistema superexcitável, porque no GABA, que é inibitório, não vai estar funcionando. Uma das primeiras publicações sobre esse assunto começaram lá no final da década de 80, então até um pouco recente. Um dos principais pesquisadores é o Ben-Ari. E aqui tem alguns exemplos de situações que causam esse tipo de crise com relação à reversão ao GABA. Por exemplo, traumas, que podem ocorrer até mesmo na vida adulta, podem fazer com que, temporariamente, o NKCC1, que é o importador de cloro, entre dentro da célula, fazendo com que o sistema fique mais excitatório. Outro exemplo é a síndrome imune materna. A gente escolheu trabalhar com o giro denteado. Nem todas as regiões do cérebro têm essa história da reversão. Algumas regiões têm, outras não têm. E aí, o giro denteado é uma delas, e é uma região do hipocampo que muito se fala sobre as epilepsias em geral, né? Então, a hipótese do nosso trabalho é saber se a infecção do Zika logo após o nascimento vai inibir a reversão a concentração de cloro no hipocampo, nos neurônios hipocampais. Esse aqui é a nossa linha, né, metodológica. Eu faço a injeção do Zika um dia após o nascimento. Então, a gente coloca 10 a terceira solução de Zika no ventrículo do animal. Então, a gente faz no ventrículo, só em um lado. E eu vou fazendo acompanhamento de pesos do primeiro dia até o 16º dia de vida, que é quando eu faço o encerramento do experimento. Também fazemos os testes de reflexo comportamentais, para ver como ele está se desenvolvendo, e o current clamp record, né, o clampeamento de corrente, que seria um PET clamp. E também ocorre a partir do... Ficou um pouquinho fora aqui, mas a partir do 10º dia até o 15º, em uma parte dos grupos, colocamos a bumetanida. A bumetanida, ela vai inibir o NKCC1, que é o que está sendo mais expresso, né, na parte do desenvolvimento, ainda na parte embrionária. E se a gente tentar inibir o NKCC1, talvez a expressão do KCC2 consiga se sobrepor e fazer com que essa reversão para inibitório aconteça. Então, esse aqui são os testes de reflexos que fazemos. O palm grasp é aquele reflexo bem clássico do nascimento, que assim que eles nascem, eles já conseguem fazer, que é de agarrar. A gente coloca algo fino, né, já que eles são muito pequenos, e eles conseguem fazer esse agarrar. Tem o recruit recovery, que é quando a gente coloca eles com o ventre para cima, e eles devem voltar à posição com o ventre para baixo, sem estar com nenhuma pata mal posicionada. Cliff aversion é uma aversão ao precipício, né, que ele não... Muitos animais, quando eles já estão com o Zika, que eu vou mostrar mais para frente, eles não têm essa noção de profundidade, então eles só se jogam. A geotaxia negativa é um experimento que vai avaliar um pouco do equilíbrio deles, onde a gente coloca numa prancha a 45 graus, e eles têm que se equilibrar, porque a gente coloca eles olhando para baixo e tem que dar a volta, posicionando-se novamente para cima. E a vibriça, a colocação pelas vibriças, fazemos um estímulo em um dos lados e suspendemos o rabo com as patas traseiras, e ele tem que se locomover na direção em que foi estimulado. Todos esses testes de reflexo vão ver como está a maturação do sistema dele. Então, aqui já é o resultado, que a bumetanida ela melhora parcialmente, pelo menos, os reflexos motores, a performance dos animais. Como eu disse, eu faço uma avaliação do comportamento a partir do 7º dia, pouquinho antes disso, muitos desses reflexos eles não conseguem fazer, está meio imaturo. Então, aqui no 11º dia, a gente vê os animais, que são o grupo controle, e o grupo que é o Zika mais a salina, e o grupo que é o tratado, que é o Zika mais a bumetanida. Quando o grupo não é tratado, que é esse, quanto mais escuro é a porcentagem da média dos animais que não conseguiram realizar. E quanto mais claro, é porque os animais conseguiram realizar. Aqui é a relação à média, e cada linha dessa é um dos comportamentos. Às vezes acontece, se vocês olharem no controle, do animal também não conseguir fazer, porque não são testes necessariamente fáceis. Mas aí, quando a gente compara com os animais do grupo Zika, enfim, a gente vê que tem essa diferença de performance. Com relação à sobrevivência, o peso deles, a sobrevivência, quando a gente faz o tratamento com a bumetanida, melhora cerca de 25%. Então, os animais, quando eles possuem Zika não tratado, eles morrem metade. Enquanto o grupo controle, todos sobrevivem normalmente. O peso, a gente vê que ele vai aumentando normalmente, sem apresentar nenhum sintoma, até o 11º dia, e aos poucos, gradualmente, ele começa a perder esse peso. E aí, quando vocês olham aqui, né, aqui é a média. Aqui tinham 9 animais. Todos esses 2 gráficos foram feitos com 10 animais em cada grupo. E a gente tem essa subidinha, por quê? Porque do dia 15 para o dia 16, 4 animais morreram. Então, a gente acabou perdendo o número de pesos que teríamos ali. Aqui é uma comparação para falar uma estatística daqui do dia 12 dias pós-infecção. Correlacionando, mostrando que tem significância do grupo controle, que não recebe bumetanida, com o grupo Zika, que também não recebe, e que não tem uma correlação do grupo controle com a bumetanida e o Zika com bumetanida. Não tem estatística relevante. Também fiz a separação desse mesmo grupo. Aqui eu não falei, né, mas nessa parte aqui já tem 30 animais nos grupos não tratados, tanto no controle quanto no Zika salina. E nos outros eu tenho 13 em cada, a gente tem um número menor ainda. Mas quando eu faço a separação dos controles entre macho e fêmea, a gente vê que não tem uma significância entre o macho ele é mais infectado que a fêmea. Não, é igual. Acho que não sei se vai dar bem para ver, mas a gente fez algumas imunofluorescência para ver como é que estava a atividade. Estava infectando ainda o Zika, e a gente consegue identificar o Zika até o 12º dia. A gente ainda consegue ver que é... Obrigada, Cláudio. Que é essa marcação verde. Onde vocês estão vendo verdinho, é tudo Zika. Já a parte vermelha é a microglia, marcando com IBA. E aí, eu não botei aqui, mas vocês podem ver, é que quando o Zika está infectando, a gente vê que a microglia possui aquela forma mais arredondada, de quando tem uma infecção ali acontecendo. Para mostrar para vocês que o cérebro também tem uma diferença. Aqui não são perfundidos, foram cérebros tirados na hora, e ele tem, em alguns animais com Zika, atrofia no cerebelo. A cor também muda bastante, não está perfundido, e ele tem esse tom mais amarelado. Bom, aqui já são os resultados em relação ao PET clamp. No grupo controle, quando eu coloco a isoguvacina é um agonista seletivo do GABA-A, que é o canal iônico que vai fazer toda aquela troca do cloro que eu estava mencionando. Quando a gente coloca a isoguvacina, por ser agonista, no animal controle, ela inibe completamente. Né? Não tem, basicamente, potenciais de ação aqui. Já no grupo com Zika, que não é tratado, aqui a gente não está falando de nenhum animal que é tratado, ele vai apresentar os spikes, porém, tem um aumento. Aqui eu contei, tem 15 e aqui tem 20. E outra coisa importante também de se observar é que tem um aumento da resistência. Quando a gente coloca, né, o step mais negativo, a gente vê que tem um aumento da resistência. Bom, por fim, esse aqui é um IV, né, um current voltagem. E o que a gente observa aqui, principalmente, embora eu pegue momentos em que a célula já está estabilizada, né, eu pego 300 a 400 de corrente, e a gente vê que a resistência até que parece igual. Mas a principal mudança é a diferença da membrane potential, onde com a isoguvacina, a gente vê que no grupo controle ela fica mais negativo, hipopolarizante. Já no Zika, com a isovacina, ela tem um comportamento de ficar mais positivo. Bom, é isso. Quero agradecer ao pessoal do Laboratório de Desenvolvimento Neural, que eu faço parte, aos laboratórios que também ajudam na pesquisa, que é a minha orientadora, e equipe de biotécnica, corpo técnico, os membros do meu CA e os docentes fora do ICE, que também ajudam nesses experimentos. Hoje eu vou estar com um pôster lá fora, então também, se vocês quiserem ver mais resultados, a gente tem mais coisa para mostrar. Obrigada.
Very nice talk, Bruna. We have time for 2 questions.
Não, porque assim, o GABA ele vai ser um antagonista o isovacina vai ser um antagonista do GABA, agonista. Então, se o GABA ele já está inibitório, ele vai tender a se tornar mais inibitório. É por isso que, tipo, como a gente acredita que, botando a isovacina, ele vai potencializar aquilo que está acontecendo. Então, se ele está inibindo, ele vai inibir mais. E se ele está excitando, a gente espera que excita mais. Mas aí a gente não pode, por enquanto, falar, realmente está tendo uma excitação, né? Porque tem várias coisas que podem estar interferindo. Exato, porque o método que eu faço é whole cell. Mas ele pode, porque...
Bruna, obrigado pela palestra. Por que você chama de Zika cell e que célula é essa?
Sim, desculpa, não mencionei. Eu faço no giro denteado naquela camada de células granulares do giro denteado. Então, meu objetivo é pegar sempre células imaturas daquela região. Mas aí eu só consigo saber se ela é madura ou não quando eu estou, né, já fazendo registro. E aí, por que Zika cell? Porque é um de animal Zika. Então, é porque como a gente não usa uma fluorescência para saber se aquela célula em si está infectada, não dá para saber. Entendeu? Talvez eu possa ter, mas a gente não... Célula glandular, na região ali do hipocampo, no giro denteado sempre. E aí a gente tem que manter esse padrão. Não é interneurônio.
Bruna, thank you so much. Now, the last guest of our session is Mikaela dos Anjos. Mikaela, she's a veterinary, and she's doing postdoc here with Professor Richardson Leão. Welcome, Mikaela. I'm really sorry. She is doing PhD. I'm sorry.
Hi. So my name is Mikaela. I am from the Neurodynamics lab, and I am a PhD student here. And I will give a short presentation here about a molecule, a new molecule we are working with. So it is a characterization of synthetic analog of psilocin. We call that UL001. And we are doing analysis of chemical, electrophysiological, and behavioral analysis in murine models. So we are going to talk about psychedelics, and I will give a short introduction about psychedelics. We find these molecules in the nature. We can find them in animals. We can find that in mushrooms, plants, and also we can find these molecules as synthetic compounds, as LSD. And it has gained too much attention recently because it's calling attention for scientists and for the medicine because it has the capacity to treat mental illness as anxiety and depression, for example. And I will give attention for this guy here, is psilocybe cubensis. You can know by magic mushrooms, maybe. And it's called psilocybe. Maybe you heard this name. And this mushroom produces this molecule called psilocybin. And psilocybin is a psychedelic molecule, well known. And once enter the body, it will be break and will be turned in psilocin. And psilocin is the active metabolite of psilocybin. So psilocybin, when it gets into the blood, it can bind some serotonin receptors. So maybe you will find that psilocybin is very similar to serotonin, and that's why it can bind serotonin receptors. And here we will pay attention on this receptor. It's the 5-HT2A receptor. And this happens in the brain, and then in the brain we can see a lot of effects like neurogenesis and good benefits for people with anxiety, for example. But we are not working with psilocybin directly. We are working with an analog of psilocybin. We can also call an analog of psilocin. So I cannot reveal the structure of the molecule, it's confidential, but we will call UL001. And it's a synthetic compound. It means it was chemically produced. And maybe, and that's why we are doing this work, it has a potential to be to have psychedelic effects. And because of that, because of it has this potential, it could be a potential new treatment for these diseases when we are talking about mental illness. And also, right now, we are already having these therapies we call psychedelic-assisted therapy. We even have this type of research here in ICe-UFRN. And our objective is to do a characterization of this molecule. So we are going to do a profile of this molecule, searching for the chemical, electrophysiological, and behavioral profile using murine models. So for the electrophysiological characterization, we are going to use brain slices of mice. And we aim to do single-cell patch clamp. We are going to focus on this region of interest, the hippocampus. And when we apply this drug in the cell, we are searching for neuronal alterations that we may see in this case, and may be similar to psychedelic effect like psilocybin or psilocin. For the behavior characterization, we are going to use this approach, is the head twitch response. It's like when a dog is wet and he shakes, it's the same behavior. So he goes side to side with his head. And it's a marker of psychedelic effect. And we are using this reference to do this work, as they use psilocybin here as a control. They were looking for another molecule here, but we can see that psilocybin produce a lot of head twitches in a long time. So we expect to see this same effect with this molecule. And also, we are searching for some correspondence with the 5-HT2A receptor, because it already have been established that this response is related to this receptor. By this research was made by applying an antagonist. So when they applied this antagonist in the mice, they have the response was distinguished. So it was not seen anymore. So when you have the antagonist, you don't have the head twitch response, which in this case means it's related to the 5-HT2A receptor. So this is our setup for the head twitch response. So we do small surgery in the head of the mouse. So we do a small incision in the head, and then we glue a magnet in his head. And we put these mice in this coil wire, and it creates a electric current, and then we can catch the signal. And it looks like this. So I have this example of what head twitch looks like. And according to the reference we have, we can do a very accurate analysis when we compare the video, the images of the video, with the signal we catch. So we are looking for that too. We already have the chemical characterization of the molecule. It was synthesized chemically, as I told you, and this the identity was confirmed by HPLC. So, my conclusion for you is that it's very important to explore this new world of psychedelics. It's not new, it's very ancient actually, but researchers are researching this right now, and it's important to explore even synthetic molecules, as we are living right now mental illness pandemic, I would say. And maybe this would be very good for humanity. And here, with this molecule, we can compare with other psychedelic molecules as psilocybin. We can compare advantages and disadvantages regarding the effects. We know that psychedelics cause some effects that are not so good for some people, but sometimes is necessary. And also, it's very important to study this molecule because we have to develop this new treatment. It's very important to go for it and treat this cause worldwide. This is my references, and this is my acknowledgements. I thank everyone that has helped me. I thank the UE Labs, which has gave us the molecule, the Hearing and Neuronal Activity Lab, which is very important for me, and my lab. I also thank a lot, and the Brain Institute. I thank a lot too. Thanks.
Thank you, Mikaela.
Sorry, I didn't hear. It's a sign. we don't know very much about how it works, because we don't know what really the mouse is feeling, you know. But we just know that he does that when he's in the psychedelic phase, the hallucinogenic phase. We know that the psychedelic is doing this effect. This effect.
No, I think it's one of those things that were observed, right? And nobody, I mean, so it's
Yeah, in the literature, it's the most response search.
no, but I mean that's the point. Like what Kia is saying, that it's a direct serotonergic effect, but you're saying that, I mean, like it's, I think it's generally accepted that it's a sign of hallucination.
Yes.
I was just thinking about the head twitch. I didn't know. I thought the head twitch was more like head banging, but it said it's side to side, right?
Yeah, it's,
And,
it's like a wet dog shake.
Maybe then the like placing the neodymium in the that is like, you know, in a parallel direction of the magnetic field. The detection is worse than if we play sidewise, because it'd be more orthogonal to the way coil is spin, you know. Because I would think like if it was head banging, then you should go up and down in the coil, right? But if you go up and down the coil
But,
to displace the thing,
like
you should put
this,
them
not like
in
this?
the side. Yeah, but then we should twist in the magnetic field, the coil roll. I just think the sensitivity of this.
Yes.
For head direction.
Yes, but we do not shake
And
our heads in the
do they
psychedelic
shake their
experience. No. Actually, I never seen that in a human, but
No, because finding an endophenotype for hallucination in rodents is the challenge, right?
yes.
And
yes,
the
yes.
question, the first question, touches to a point that's very important, because you may be measuring actually a side effect of activating serotonergic
Yes.
circuits.
Yes, it's
And it's a proxy. We all understand that we work with rodent models of disease,
yes.
right? But the goal is to go for that direction to try to get the closest endophenotype to what we call an individual's subjective experience.
Yes.
Right?
Yes.
I
sure.
think that's the goal.
Yeah.
But I think it's still a trajectory.
Yeah, I agree. And maybe right now, this is the only marker for us to know that the animal is having a psychedelic experience. But we don't know, in fact, what it's feeling, what he's experiencing, right? But you are right. Thanks.
Thank you so much, Mika. Now we are gonna invite you to join in our coffee break and also to poster session, and we will be back at 11:15. Thank you.